So protein kinase reaction is because of the catalytic center of the protein kinase, and that is mainly due to the acidic amino acids, two metal ions, and then basic amino acids that are present in the catalytic site of the protein kinase. So basic amino acids, acidic amino acids, along with metal ions, mainly influencing the stabilization of the structure. So I was asking you that the transition of closed structure to open structure. So the structure of protein is very dynamic. Okay. So any of the factor, yes,
Sir,
Sangeeta.
so there is something on the screen, sir. Square, rectangle, sir.
Yeah, what is this? I
Black.
don't. Okay, good. Now it is fine, right?
Yes, sir.
Okay. So conformation of the protein, please do not forget. The conformation of the protein is very important. Understood. So when the phosphorylation is taking place, phosphorylation definitely, that is the point exactly Pooja said. Phosphorylation is the one which is responsible for the activity change. But what happens if phosphorylation is taking place, right? What is happening? Conformational change, because I said that protein is a charged molecule and imparting negative charge within the protein influence change in the electrostatic interactions, thereby conformation will be altered. Understood? So what is happening if conformation is changed? So the catalytic site, activation site will become open. That is what open structure. When it opens, it become again accessibility. Like in case of histones, nucleosome structure we were discussing in molecular biology. Here also, when catalytic site is open, then substrate can actually bind to the catalytic site where the phosphorylation is taking place by this protein kinase. Understood now? So that is what I expected. Conformation, protein conformation. Conformation is you are confirming that you are going to write the exam today. Conformation means the quaternary structure of the protein. What conformation means? What is its native structure of the protein? That is conformation. So protein conformation, I said it is very dynamic and it is under the influence of many factors like the incorporation of charged residues like phosphoryl groups, phosphate groups, or pH can also influence, or even the temperature. Like that, many factors or any factor that influence, for instance, the change in the structure, nothing but change in the activity status of that particular protein. Understood? So now basically either acidic amino acids, metal ions or basic amino acids are responsible together for the stabilization of the ATP as well as the protein structure. Okay. So now this is what we have discussed. This is in case of the protein kinase A enzyme, and this is the activation site where all the 270 amino acids we discussed, they are conserved in the activation site, are responsible for the function as a 3 point that we discussed earlier slide. So these are in protein kinase for your exam purpose. You get question like protein kinase A activation and protein kinase reaction is this is the one. So here you have different amino acids and we discussed earlier in the last class D166. This is very, very important for the functional role of protein kinase. D means aspartic acid. So at 166 position, this aspartic acid is the one which is actually interacting with the substrate hydroxyl group. Activation. Earlier we discussed activation, right? So this activation of hydroxyl group is because of the D166 aspartic acid and it is responsible for the initiation of phosphorylation of the substrate, right? So carboxylate group of aspartic acid functions as a base and activate the hydroxyl group of the acceptor serine for nucleophilic attack on the gamma phosphate of ATP. So these three here are very important in the first reaction. You have to have the substrate that is another protein kinase, as I said, which is undergoing phosphorylation. And these are serine threonine tyrosine. So they have got hydroxyl group on which phosphorylation is taking place. Then your enzyme, this is enzyme at catalytic site D166. This group, carboxylate group of aspartic acid, is influencing or initiating this phosphorylation reaction. And another one is ATP, which is a phosphate group. All this in the first step is initiated by the D166 residue. Then N171, N is for which amino acid you need to find out. So this is responsible for hydrogen bonding to D166. Then D184, that part of DFG motif yesterday in catalytic site, you have DFG tripeptide motifs. So DFG motif in the activation group is responsible for Mg2 plus binding. 184, this is the 184. It interacts with metal. Earlier slide we discussed metal is having important role in stabilizing this gamma phosphate. So here you can see this is the second step. First step is activation of hydroxyl group. Then second step is through the metal ion by the D184, which is present in the DFG motif, stabilizes this ATP phosphate group. Then K72 orients alpha and beta phosphates of ATP. So this K72 from some different position is responsible for proper orientation of ATP in such a way that the ion pairing with E91. So these are all things are responsible for the donation of cleavage of gamma phosphate and adding this gamma phosphate to the substrate. So E91 in C helix. So that is E91 is present in the C helix is ion pairing with the K72. We have discussed earlier in the structure that there is a salt bridge formation in case of C helix. So this C helix is responsible for regulation. How? Through this. So mainly this protein kinase reaction, whatever the phosphorylation of the substrate, is mainly influenced by the salt bridge in a way that stabilization of the structure during its function. Understood? So that is how the different steps here, starting from first very important step of activation of hydroxyl group, then forming the hydrogen bonds to D166, and then the other groups finally responsible for stabilizing ATP and the protein kinase structure so that exactly gamma phosphate is removed and added to the substrate. Understood all of you? So what are the factors now that makes the substrate specificity of this protein kinase? And this is determined by the sequence of the phosphorylation site and other structural part of the substrate protein. So within the substrate protein kinase again, the moieties, that is serine threonine and tyrosine moieties, which are undergoing phosphorylation, that are going to determine the specificity and docking of substrate protein. So docking means so here enzyme is coming in contact with the substrate, that is docking on the receptor tyrosine kinase. This is example. So docking of protein on receptor tyrosine kinase via SH2 phosphotyrosine interactions. So this is the way how it is interacting is deciding the specificity of phosphorylation. Understood? So the interaction helps to clamp the substrate onto the kinase, ensuring high efficiency of phosphate transfer. So what is this point? How this point is giving the inference of specificity of protein kinase function. So specificity, how specific protein kinase is functioning depends on the way how the substrate is interacting with the catalytic center of the enzyme. Understood? So that is what here. Docking means two things are coming in close contact. So here, what are the two things? One is in this example, receptor tyrosine kinase, and it has got SH2. If you recollect, we discussed about SH2 PDB domains, right? So this is phosphotyrosine and SH2 moiety interactions. They are very critical in determining the specificity of phosphorylation site. Then another factor is interaction modules distinct from this phosphorylation site. The phosphorylation sites are specific based on which classification is done. So apart from that, there are some other interaction modules which will again contribute for the specificity of the substrate phosphorylation. Another one, co-localization of protein kinase and its substrate can enhance the substrate specificity. So localization is very important factor in signal transduction because after its activation, one important signal or event in the signal transduction is translocation, for example, like from cytosol to nucleus. Whether the phosphorylated protein is in the nucleus or cytosol is an important indication of its activation. Understood? Same way here, the specificity of that particular substrate getting phosphorylated also depends on the co-localization, means both getting localized. What both kinase engine and substrate. That is also very important that determine the substrate specificity of this phosphorylation of the protein kinase. So protein kinases can act. Understood? That is all about the protein kinase reaction and specificity. Now next topic is how this control is taking place, this activity. So mainly protein kinases in that way it is acting as a switch, activating or inactivating. That's why it is acting as a switch in the signaling pathway. So different activating and inactivating signals, whether signal transduction pathway has to be activated or inactivated, decided based on activation and inactivation signals, positive and negative signals in the cell that can alter the activity through change in the conformation as we discussed. Mainly proteins, they whether they are active or inactive is depending on the conformation, not conformation, conformation. So the protein kinase structure contains several flexible elements that comprises the hinge region between the two lobes. In the structure we discussed. So the portion that is present between the small lobe and the larger lobe, that is the flexible elements, and the activation loop, the P loop, and the C helix. So these are all the regions present in between the two lobes through highly coordinated movement during transition between on and off state. So these regions are mainly responsible for controlling the protein kinase activity. Then activated state of protein kinase exhibit following characters. What are the characters that are exhibited by the activated protein kinase? So two lobes are closely packed together. Then ATP is buried between the two lobes and its phosphate contact the P loop. The C helix is positioned for salt bridge formation to a beta strand of the N-terminal lobe. Okay, so this way the changes from inactive to active state is taking place. Then the binding site of the peptide substrate and ATP are fully accessible. Mainly accessibility is nothing but the activation state is associated with. So phosphorylated activation loop has moved away from the active site and adopt an extended conformation. So the loop helps to organize the catalytic residues for optimal phosphate transfer, and it forms part of the substrate binding site. So movement of the activation loop is often coupled to a movement of the C helix. So this way C helix is very important that controls the changes. Then inactive states are characterized by the uh, tightly, this is not actually open conformation. Okay, this is not open conformation. Open conformation is for activation state we discussed. No, two lobes are closely packed in the activated state. Two lobes are relaxed. This can be relaxed conformation of the two lobes. Understood? Then precluding an optimal orientation of the residues involved in substrate binding and catalysis. So these are the factors responsible for inactive state of the protein kinase. So next I can continue in the next class. If any questions for the present class discussion, I can answer. All of you have followed, so you have to read this material and go one by one, then practice to remember. What are the different changes associated with conformational change or activation state of the protein kinases? What is protein kinase reaction? What are all the different moieties involved in protein kinase reaction? That is phosphorylation of the substrate protein kinase. And then what are the different ways of this regulation? That's what we have discussed. So still some more slides are there. I can continue in the next class. But for the present class, I wanted to know if there are any questions. Because you will get confused. There are so many new terminology and concepts. Any questions you have? Any understanding, misconception still not clear? You can talk to me. So there are no questions because you think now like you are following, but it's really not getting into clarity of understanding in your brain, right? So for that, you need to again read, stay some time for each and every point, try to understand, and then practice. Got it? So if there are no questions, I am stopping here. Time is also over. Then today afternoon, 2 o'clock, your exam. See you all.